Assalamualaikum warahmatullahi wabarakatuh. I am Muhammad Hafiz bin Wahida from AS114-F5. Before we start our presentation, I would like to introduce my group members, which is Muhammad Qayyim bin Ahmad, Wan Muzamil bin Wan Mushta, Wan Christina Batrisha bin Wan Ahmad Fahri, Putri Nun Hanadin Najira binti Nur Hash Hamadi. So, I will start our presentation about metabolic interrelationship. So, what is metabolic interrelationship? It is known as metabolic integrated metabolism. It is central array of metabolic pathway that exists in all living organisms. It is used to break down most important molecule in the body known as ATP, and a lot more to generate energy for basic living things. Let's take a look at in our body. Every organ has their specific works to do to maintain the metabolic cycle. For example, liver function act as central hub that supply glucose to the brain. In human body, this concept is important to understanding to help us know better about diabetes, starvation, and psychologist exercise.
Next, I will explain about the biological function in cell. The first function is homeostasis. The primary function of a cell is to maintain a stable internal environment despite the external changes. Metabolic interrelationship allow the cell to adjust its chemistry instantly. So, how does this homeostasis work is if blood sugar drops, the cell's metabolic relationships shift as it stops storing glucose from glucogenesis and start breaking down stored fats or proteins to maintain the concentrations of ATP needed for survival. In this way, the cell can avoid starvation as there is always ATP needed. The next function is detoxification. Detoxification is a biological function that produce toxin byproducts like ammonia from protein breakdown. Relationship allows the cell to neutralize toxic nitrogen waste into urea while simultaneously generating energy producing intermediates. Without this link, the cell would essentially poison itself during protein metabolism.
Let's take a look at ATP or adenosine triphosphate. ATP is what the cell uses in our body to keep things stable. And to explain this furthermore, let's imagine that ATP is like a battery. The battery or ATP is made out of three phosphate group, and in anabolism, the third phosphate group bond is break via hydrolysis, and this creates energy. Now, the ATP is converts into ADP. There is adenosine diphosphates, or in other words, the battery is now lost its energy value. Now, in catabolism, the cell breaks down the glucose to get the energy, and then this energy is used to create the bond between the ADP and the loose phosphate group to make the ATP. Or in other words, it will recharge the battery to become full. Before going back to the poster, let us take a look at the difference between catabolism and anabolism. Catabolism use complex molecule to make a simple ones. This is an exergonic or it produce ATP. The goal of catabolism is to release energy. In other hand, anabolism use simple molecule to make a complex one. It is endergonic or it use ATP. The goal of anabolism is to building a biomass. Now, with this understood, let us take a look at the poster. Now, let us take a look at the biological function in cell or the flow. First, carbohydrates undergoes glycolysis in cytosol to become glucose. The 6-carbon glucose molecule is snapped in half to create two 3-carbon molecules called pyruvates. This produce 4 ATP, but it's invested the 2 ATP. Now, the second flow is the fat. The fat undergoes beta oxidation to become the fatty acid. Fatty acids have 16 carbons. The 16 carbon is then clipped two at a time and then become acetyl-CoA. Or in other words, two molecule of carbon from the fatty acids equal to one acetyl-CoA. Now we go to the third flow. There is the protein. The protein undergoes deamination to become or break down to amino acid. Amino acid contain nitrogen. The cell remove this nitrogen into the waste that we call urea or urine. Depending on which amino acid it is, the rest of the molecule now become pyruvate, acetyl-CoA, or it can jump directly into the Krebs cycle. Now, let us take a look at the carbohydrates and protein that have been converted into the pyruvate. The pyruvates become acetyl-CoA via the process we know as pyruvate oxidation. The acetyl-CoA enter the mitochondria and then undergoes the Krebs cycle to produce 2 ATP. Then they undergoes the ETC or electrons transport chains to produce 30 or more ATP to keep our body alive.
One of the manufacturing industry is widely applied in making antibiotics, enzymes, and vitamins. This is an application for industrial fermentation of penicillin production. Metabolic interrelationship refers to the close connection between different metabolism pathways such as carbohydrates, protein, fat, and energy metabolism. In the production of penicillin, microorganisms like penicillium are grown in large fermentation tanks. These microorganisms use carbohydrates such as glucose as a primary energy sources. The energy released through carbohydrate metabolism to support cell growth and activate other pathways. At the same time, amino acid metabolism provides essential building blocks required for the synthesis of penicillin. The coordination between energy metabolism and amino acid metabolism ensure efficient production of the antibiotics. By carefully controlling nutrients, temperature, oxygen supplies, and pH, manufacturers can regulate this metabolic interrelationship to increase yield and efficiency. This application is essential in producing antibiotics, enzymes, vitamins, and bioproducts on a large scale.
Why insulin is manufactured? Insulin is a hormone used to treat diabetes. Large scale of insulin is produced using biotechnology because animal-derived insulin is limited and may cause allergic reaction. Therefore, recombinant insulin is more safe, more pure, and more consistent to be used. E. coli or yeast are the example of microorganism used and will act as a biological factories. Insulin production depends on interconnection of several metabolic pathways inside the host cell, which is carbon metabolism, amino acid metabolism, and energy and redox balance. For example, If glucose metabolism is optimized, more energy is available on insulin synthesis and may help reduce metabolic stress on the cell. This will ensure efficient insulin production and may help reduce the manufacturing cost.